Hi everyone, welcome back. This particular video will be longer than normal just because I'm going to go through the lab manual pages 141 through 173 in more detail than normal, mainly because we're going to get into a lot of Punnett squares, and for some of you Punnett squares are a little tricky. So I will actually be solving a lot, some of the Punnett squares with you. So for those of you that have Punnett squares down pretty well, feel free to skip through parts of the video and just go and do it on your own, okay? So as always, go ahead and download the supplement directions. Luckily this week for the supplement, most of it in the lab manual, for the first part of the lab manual, it's basically just skip through. You basically just follow the lab manual part. The only place that you really need the supplement is for the second part when we get into Mendelian genetics and the Hardy-Weinberg. Okay, so we'll talk about that in a bit. So let's get into lab topic 7 with Mendelian genetics. So for this first part, you basically will need to do some definitions. So it's a good idea to define these terms from your textbook. So looking up the key terms in your textbook, but rewriting them in your own words. So, what is a gene? What is, a genotype versus a phenotype, and so forth. But thinking about it in your own words, okay? So that will help you remember things, okay? And then, thinking about when we go over it, okay, what's a homologous chromosome versus homozygous versus heterozygous? So think about, how some of these terms, they sound similar to each other. So just realize that when we have these definitions, if we have them down really well, then we'll remember that like a homologous chromosome, those, they are basically mean that they carry the same genes, but not necessarily the same alleles of the same genes. Remember, so an allele is a variation of a gene, okay? Such as, one allele of a gene may say widow's peak, whereas the other allele means straight hairline, okay? So forth, all right? So just be aware that it's a good idea to write these definitions down and commit them to memory, so that when we go through this terminology, you understand like homologous chromosome right away is different from homozygous. All right. So as we go through, answering the questions along the way, even though some of these questions might seem easy, it's just good practice. So what is the phenotype of an individual whose genotype is big R, big R? Okay, so they are the ones that have the dominant allele. Heterozygous, they have the dominant allele. Homozygous recessive, they have the recessive allele, and so forth. Okay. Remember the different laws that we talked about with Gregor-Mendel. So the principle of segregation. So during anaphase, the idea that the chromosomes, or I should say it's the homologous chromosomes, they're separating into different daughter cells. And so that means that when we have something like big R as a homologous pair, that the resulting gametes, one gamete will have a big R, the other gamete will have a big R. Okay, so this person can only have big R's in its gametes, whereas somebody who's heterozygous, half of his gametes will have a big R, half of his gametes can have a little r. So understanding, what does this mean in relation to meiosis. Okay. When we get to the monohybrid crops, so this is when we start getting into Punnett squares. So with Punnett squares, we have to figure out basically what is a parent generation versus the F1 generation versus the F2 generation in relation to Mendel's laws, okay? With Mendel, remember the parent generation are always those true breeding varieties. So with the true breeding varieties, they're always going to be homozygous. So here we have homozygous dominant with a homozygous dominant. So if we have a homozygous dominant and a homozygous dominant, they're basically only going to produce homozygous dominant F1 generation, right? So big R, big R. What is the phenotype? They're always going to have whatever the dominant allele is. Okay? If we have something with, and you can do the same for the next one. When we set up a Punnett square, these Punnett squares are set up a little differently from the ones that I did in lecture. They're just on their side, okay? So instead of how I had it where we have like a box, it's more of a diamond shape. So you have, you know, the eggs on one side and the sperm on the other, instead of the eggs on top and the sperm on the side, or vice versa, okay? So you can see how it's already written in. We have little r, big R, or big R, little r, big R, little r. So you're going to do the same thing where you're going to bring the big R down. So big R, little r, little r. And then this one goes this way. Big R, little r, and solve it from there. Okay, so here we have homozygous dominant, here we have two heterozygotes, and here we have a homozygous recessive. Okay. In activity C, it's showing that the chromosomes, most of the time we just assume that the chromosomes are on separate, the genes are on separate chromosomes, so they are not linked. Occasionally we'll come across chromosomes that are on the same, or genes that are on the same chromosome. So that means that they are linked, okay? But if it doesn't say that in the problem, just assume that they are unlinked. So see, for example, here it says two unlinked genes are called RNA. The heterozygote is, the cell is heterozygous for both. So what is the genotype of this cell? So if the cell contains a chromosome, two homologous pairs of chromosomes, we have one pair of chromosomes that have the genes R on it, and another pair of chromosomes that have genes A on it. So, and then for gene R, it's heterozygous. For gene A, it's heterozygous. Okay, so you're going to draw a picture of that. So what would that look like? Okay, I'll draw it with a pencil. Okay, so I could have one chromosome. So here's one pair of homologous chromosomes. I'll do a different color for the other pair. Okay, so this one over here would be A. So I would have big A and little a because they're heterozygous. For this one over here, I would have big R, little r. Right? So then what is the genotype? So if you remember the genotype, it has one big R, one little r, one big A, one little a. Okay, so that's how you would write that down. All right. And then you can answer the question about what is the phenotype if it has the dominant allele for both. Okay, so now we have to figure out how would we represent that in a dihybrid cross. Okay, so if we have genotype RRAA, they're all uppercase, what would be the possible gametes? So remember with foil, first would be, with the foil we do first and then outer and then inner and then last. But that all ends up to be the same thing. So I would only need to do big R, big A. For this one, same thing, you do first. So that would be big R, big A, and then I do outer, and it's also big R, big A. I do inner, and we have something different. It's little r, big A. And then last is also little r, big A. So we have two different gametes for this second genotype. For the third genotype, the first one is little r, big A. Little r, big A. Outer would be little r, little a. Inner would be little r, big A, so I don't have to do that one again. And then last would be little r, little a, so I don't have to do that one again. And then finally, for the last one, it's going to be 4 different possibilities. Okay, so remember, if we do first, it's going to be big R, big A. If we do outer, that's going to be big R, little a. If I do inner, that's going to be little r, big A. And then last will be little r, little a. So there's 4 possible gametes there. Okay. So here, let's see. We listed all the possible outcomes here. So to predict the outcome of a cross, construct a Punnett square to accommodate the gametes from each parent. For example, cross 2 heterozygous parents using the Punnett square. So basically, here's our Punnett square. So we're using these gametes because these are the parents. So I need to move these gametes over here for the eggs and for the sperm. Okay, so here's my gametes. So I'm going to have big R, big A over here, and then here's another one, big R, little a for that box. Here's this one, little r, big A, and then little r, little a. I'm going to repeat the same thing for the sperm because we have another heterozygous parent. So I'll do the same thing. Big R, big A. Big R, little a. Little r, big A. And little r, little a. All right. So now I'm going to combine the squares. Okay, so we have, big R, big A, big R, big A. Okay, so we just move them together. We want to put the R's in the, all the R's together and all the A's together. So when I bring them together, it'll be big R, big A, big A. Big R, big R. So this one and this one come together. So big R, A, little a. This one and this one. Big R, little r, A. I like to put the capital letter first and then the lowercase letter. This one and this one. Big R, little r, big A, little a. Now I'm going to do this one and this one. Big R, little a, little a. This one and this one. Big R, little r, big A. This one and this one. Big R, A. This one and this one. Big R, big A. And then this one and this one. Big R, A, little a. This one and this one. Big R, little r, big A, little a. This one and this one. little r, big A, this one, little r, big A, little a, this one and this one, we have little r, little a, this one, little r, big A, little a, this one and this one, big A, little r, or big R, little r, little a, and then finally we have big R, big A, little a. And then list the possible genotypes of the offspring. So you can look through and kind of see that basically all of the possible offspring are accounted for. So there should be, you know, all of the different possible offspring. So you should be able to see that there's a big R, big A, there's a big R, big A, little a, there's a big R, little a, there's a big R, little r, big A, there's a big R, little r, big A, little a, and the, let's see, big R, little a, and a little r, big A, and a little r, big A, little a, and finally a completely homologous recessive. Okay, so there's all those possibilities. If you had looked through, you can kind of cross them through. Okay, and then all the phenotypes that go with it. Okay, so like this is all dominant, so these two are the same phenotypes, along with, you know, this one. So you can see like which ones are the expected phenotypes and so forth. Okay, all right. So that's that. Dihybrid cross with fast plants. I think we skipped that one, right? Yep, so we're going to skip that one. Okay, and then we'll get to the reviewing your knowledge in a second. All right, so let's go into the next one. Okay, so that gives you an idea of dihybrid crosses. So now we're going to get into the Mendelian's supplement. Okay, so we're going to get into something called Hardy-Weinberg principle. So with Hardy-Weinberg principle, we can take something called population genetics, and we can kind of predict the outcome of a population, the frequency of alleles within a population. So we're assuming that, you know, we have 100% of a population, and then within the population, if we know how many of a population of a certain gene have the recessive phenotype, we can get an idea of what the percentage might have, the how many might have the dominant allele, based on this this formula. Okay, so when we look at this formula, there's a couple of things that we'll notice. Okay, so we have, it's called it's in equilibrium. So basically it's p squared plus 2pq plus q squared equals 1. So when we look at this formula, there's a couple of things that we want to point out. So p squared equals the basically the the percentage or of the individuals that carry the that are homozygous dominant. So these are the homozygous dominant within the population. So p squared are the homozygous dominants within the population. 2pq represent the heterozygotes within the population. And then the q squared represent the homozygous recessives within the population. All right. So if we know if we have, part of this equation, we can figure out the rest. All right. So if we, so basically again, so if we have, p squared represents, the percentage of the population that are homozygous dominant for a particular trait. 2p cubed represent the percentage of the population that are heterozygous for a particular trait. Q squared represents the percentage of the population that are homozygous recessive. So if you notice, these, if this was 30%, this was 40%, this was 30%, 30 plus 40 plus 30, that adds up to 100%, right? So this would create like an equilibrium. So this would add up to 100%, or 0.3 plus 0.4 plus 0.3 equals 1. So this, is an equilibrium here. So we're going to play around with this equation. Now, when we're looking at this equation, the P represents the dominant allele. The Q represents the recessive allele. So if you look at this, P squared, that represents the homozygous dominance. 2PQ, that represents, the people that have the ones that have both the dominant allele and the recessive allele. And then Q squared represents 2 recessive alleles. All right. So, so we'll play around with that in a minute. Okay, so let's, here it says, if the relationship between P and Q remains constant through random mating generations, the population is said to be in Hardy-Weinberg equilibrium and no evolution occurs. In order for a population to remain in Hardy-Weinberg equilibrium, several conditions must be met. So list them from your text. So we actually haven't gotten to this in our text, so I'm just going to talk through them. So the first one that makes it so that we're in Hardy-Weinberg equilibrium is that we must have a large population. Okay, so this works if we have a large population. If we're only dealing with like 3 people, this isn't going to work. Okay, so we have to have a very large population in order for us to, make sure that we have enough random mating and all, this Hardy-Weinberg equilibrium to be met. Okay, so it's not going to work if we are dealing with a small population. Another condition that must be met is that we have random mating take place. So if people are choosing their mates, or if individuals are choosing their mates, it doesn't work either. So every individual has to have an equal chance of reproducing in order for this to work. So right away you can tell that this Hardy-Weinberg equilibrium has some issues, okay? Obviously this doesn't really work well for humans. The third condition that must be met is that there's no gene flow within the population. So what that means is that we don't have any immigration or emigration into or out of the population. So that means that the population stays the same. Okay, so nobody is entering or leaving the population. Okay, obviously that doesn't occur normally. Usually within any population, even a squirrel population or a bird population, somebody's moving in or leaving the population at any time. Okay. The fourth condition is that no mutations are taking place. Okay, obviously that's something that, we can't control. There's always some sort of mutation that would be taking place. And then finally, no natural selection. Okay, which of course, again, we don't have any control of either. Okay, so you may be wondering, okay, wait, these 5 conditions clearly, other than the first one, clearly cannot be met by most populations, so why are we even learning about this Hardy-Weinberg thing? Well, it's the closest that we have. So, nature is, it's uncontrollable. So this is, the Hardy-Weinberg equilibrium is basically the only thing that we have to get like a clear glimpse into how we can kind of see how populations change over time. So even though, this is not an exact science or anything like that, we can kind of use the Hardy-Weinberg equilibrium just to kind of, get us a guesstimate to see if, we can get into a glimpse of, percentage of the population for each version of a gene. Okay, so it's not perfect, but it gets us what we need. All right, so those are the conditions. Obviously, we can't make them all, but we can get pretty close, or get close enough for our purposes. All right, so you are going to perform a test at home. So your lab kit actually contains these paper testers of 3 traits. So you should have, I think you should have 4 pieces of paper, if I recall. I can't remember. One should be just a regular piece of paper that's a control. And then the other 3 have chemical groups for your taste buds. So one's a PTC paper, the other thiourea, and then sodium benzoate. So you can test to see if you can taste each chemical, okay? In between each test, you'll want to actually this is misspelled. You want to rinse your mouth in between each test. So just take a drink of water. Some of you might have a strong reaction to the paper, okay? So just be aware that you might have a strong reaction. So you might be what's called a hyper taster, okay? Some of you might not taste anything, it just tastes like paper, okay? So for example, I am a non-taster when it comes to PTC paper. So when I do this in-person tasting, I like to do this in front of students. I'll take the PTC paper and I'll put it in my mouth. And to me, it just tastes like paper. But then when students put it in their mouth, half the class is just utterly disgusted and they're like, ugh, this is disgusting. And they're spitting it out. And I'm sitting there going, what is the big deal? Okay, so, just realize that if it just tastes like paper to you for any of these 3, you are a non-taster, okay? You will know if you are a taster, okay? You will know. I can't describe it because I am a non-taster for actually all 3 of these because I don't taste anything. Okay, so I don't know what they taste like, but from what I can tell from the other students' reactions, it's pretty distinctive if you are a taster. Okay, so if you are a taster, you will know, and you can, write down, just put a check if you're a taster or not. The control paper is just a piece of paper, so you can taste what paper tastes like, so you'll know if you're a taster or not, okay? Since you won't be able to know what other people tasted or not, we gave you some just data that you can write down in the data table there, okay? And then from there, you're going to take this class totals and you're going to write it as the Hardy-Weinberg equilibrium, okay? So to do this part, think about, so the non-tasters are basically the recessive traits. So if you are a non-taster, we know what your genotype is, okay? So if you're a non-taster, you are Q squared, okay? So the non-tasters are Q squared, all right? So that means if we take a look at the number, okay, so if you are a non-taster, and let's see, how many non-tasters there were 5 of you, okay? Out of 20, 5 plus, 15, there are 20, okay? So if there were 5 non-tasters and 15 tasters, so we know that, there's 5 divided by 20 equals what? Okay, so that equals 0.25, right? Okay, so that means that Q squared, I'm just going to do a caret for squared, because I know that Q squared, that's the non-tasters, that's the homozygous recessive, is 0.25. So how would I figure out what Q is? How do I figure out what Q is if Q squared is 0.25? Okay, so Q squared is 0.25. How do I figure out what Q is? I take the square root, right? So what's the square root of 0.25? Well, you got to do the calculator. So get the old calculator out and find the square root of 0.25. So I just figured it out. It's 0.5. Okay. So now I have Q equals 0.25. So if I know what Q is, I can figure out what P is. Okay. So P plus Q equals 1. So that means Q minus, P equals Q minus 1. So what that means is that P must equal 0.5. And then that means that 2 times 0.5 times 0.5 would equal the number of frequency of heterozygotes. So what is 2 times 0.5 times 0.5? That would equal 0.5. Oops. Let it go. 0.5. All right. And so, based on this, I should be able to check and make sure. So if I have, if I know what Q squared is, so remember our equation going back up here, P squared, 2PQ, Q squared. So I know Q squared is 0.25. I know P squared, or 2PQ is 0.5. So what must P squared be? Okay, it must be also be 0.25, right? In order for it to equal 1. Okay, so you can always use the equation to kind of make sure that your numbers make sense. Okay, so I solved the first one, you have to solve the other two. All right. Okay, so in 7.2, you're going to be investigating the possible student phenotypes and genotypes. Okay, so this is just given based on the dominant and recessive forms of each trait for yourself. So list the possible genotypes and phenotypes given. Okay, so in our previous lab, up previously, it talked about freckles and stuff like that. So think about yourself, okay? Do you have freckles? If you do have freckles, was that a dominant trait or a recessive trait? If can you think about what your possible genotype is, and what your possible gametes would be, and so forth? You can leave out the group for that. So all that group stuff you can just leave out. Astigmatism, so whether you have an astigmatism or not, if you don't know if you do or not, if you've ever really like focused really hard on something, so if, for example, if you take out like a medicine bottle that has really, really tiny writing on it and tried to read it, and then you looked away at something far away and tried to read that, and it was really blurry for a while, that means you have an astigmatism. So for example, I have an astigmatism. If I look through a microscope, and this happens a lot like when I'm teaching in-person labs, I'm looking through microscopes for students and I'm really focused in on whatever, you know, is magnified, and then I look up. It really takes a while for me to like focus, refocus on like normal everyday stuff. Because of the astigmatism. So if you're really focused on really small stuff and then you suddenly look up and try to refocus, that means that you have an astigmatism, and if it's really blurry after a while. So if that's a yes for you, then you have an astigmatism. If that's no for you, you have normal vision. And you can figure out your possible genotypes. Tongue rolling, so if you can roll your tongue or not, figure out if you're yes or no for that. Arches or flat feet, yes or no. Widow's peak, if you have a straight hairline or if it comes to like a little point. Okay, so you can kind of play around with that and then answer the questions. All right, going back to the supplement, we do have a video on how to complete the blood type. So in your test kit, you have the Eldon card blood typing kit, so you can actually figure out what your blood type is using it. So with the blood type kit, you can, well, here's some background information about blood typing. So you can see like the prevalence of the different blood types around the world. Here in the United States, we're kind of a melting pot. So we're pretty well represented for our blood typing. O is the most common. I think it's about like 40% of the population are O. But as far as like A versus B and so forth, you can see like the B allele tends to be a little bit more common, you know, in Asia. A tends to be a little bit more common in European descent. Africa is, you know, pretty well represented for both. You can kind of see like, as far as everywhere else, it's pretty well spread throughout. So there's no really dominant allele anywhere. We're pretty well spread out everywhere. So you can read the information about like what the RH factor is and so forth. So when you're ready, you can perform the test to see what your blood type is. So you can follow the YouTube video. So the YouTube video just kind of walks you through how to take the test. If you already know what your blood type is, you know, you don't have to do it again if you don't want to. If you really don't want to, you know, take your blood, you don't have to. I'm not going to like force you to, but it's always, you know, good to know what your blood type is, especially when you're going into the health sciences. If you've ever donated blood, you should know what your blood type is. If you've never donated blood and you want to, there's all sorts of blood drives that you can, you know, that are going on. Just go to redcross.org. They have blood drives all the time and are always in need of blood. So please, you know, donate blood. I used to donate blood all the time before I became ineligible. So I really encourage you to save lives. It's always neat because you can always, they always send you emails like, oh, you know, your blood was used at this, you know, hospital, you know, so that's kind of neat. It's like, oh, you saved somebody's life at like in Eugene or something, or in like North Dakota or something, you know, so it's kind of neat. It's like your blood got used by somebody and saved somebody's life. So, you know, save a life, donate blood. It's pretty easy. So, and then you can kind of see, you know, what your blood type is if you haven't figured it out. So there you go. So you're welcome to do that if you'd like. So this is where you record your information from the Eldon card. And then we have some data that you can transfer over from the people in the class of who whose blood type is whom. So you can just transfer that over into your chart and then answer the questions. Okay? And then finally we have exercise 7.4. So that goes into lab topic, the genetics problems, the weird practice problems. Okay, so these are just practice problems from monohybrid crosses, and then dihybrid crosses, and then incomplete dominance, and then multiple alleles, and then sex-linked questions, and then sex-linked dihybrids, and so forth. So for those of you that are pretty confident in your Punnett squares, you can do all of this on your own, and then basically you're just submitting pages 5 through 7 along with the other pages for points. But I'm going to go through some of these questions for the rest of you. So let's take a look at these questions in detail. Okay, so hang on a second. Okay, so again, I'm going to go through these with students, starting with the monohybrid cross. So we're going to set these up and go through them together. So let's start out. It says here, in peas, yellow color big E is dominant to green little e. A homozygous yellow pea is crossed with a green pea. So when I read these questions, as I read them, I start writing exactly what the genotype is. So it says homozygous. So homozygous means two of the same, right? So homozygous yellow, big E is crossed with green, little e. So those are the genotypes. What will the colors of the offspring of the cross be? Okay, so we need to set up the Punnett square. Okay, so this one's already a little different. So we're going to put one of the genotypes up here. So there's big E, and then this guy I'm going to put on the side, little e. I'm going to bring these down, big E. This one goes down as well, big E. And then this one I'm going to bring across, little e. So what will the colors of the offspring of the cross? So look at the offspring here. What color are they? Are they yellow or are they green? Well, they all have the big E, so they're all yellow. Give the genotypic and phenotypic ratios. So all of them are going to be big E, little e. So with the genotypic ratio, the way that we write it out is we write it out as the homozygous dominance to the heterozygous to the homozygous recessives for the genotypic ratios. So in this case, how many homozygous dominants do we have? We have 0. How many heterozygotes do we have? We have 4. How many homozygous recessives do we have? We have 0. So for the genotypic ratio, we have 0 to 4 to 0. Now for the phenotypic ratio, we do the same thing. We do the number that have the dominant phenotype to the ones that have the recessive. Recessive phenotype. So how many have the dominant phenotype? How many have the recessive? So if I was to give the ratios for the genotype, it would be 0 to 4 to 0. Phenotype, it would be 4 to 0. All right, so that's just how you properly do that. Now, I'm not too picky when I grade these things, so if you do, you know, they're all heterozygous, I would be fine with that, okay? Or they're all yellow, I would be fine with that, all right? A heterozygous pea, yellow pea, so right away I'm going to write the genotype, is crossed with another heterozygous pea. Give the parents' genotype. So I just did. What offspring would you expect from the following cross? Okay, so I'm going to set it up again. So we have a big E and a little e, a big E and a little e for the parents. So I'm going to bring the big E down. I'm going to bring the little e down. Big E across. I always like to do the capital letters first, you'll notice. And then the lowercase letters last. So there are my offspring. So what offspring would you expect from the cross? So I have basically 3 yellow and 1 green. Okay, so let's do the genotypic ratios first. So how many homozygous dominants? I got 1. How many heterozygotes? I got 2. And how many homozygous recessives do I have? I have 1. So it's a 1 to 2 to 1 ratio. And then for my phenotypic ratio, what do I have? I have a 1, 2, 3 yellow to 1 green. All right. Next, we have 2 long-winged fruit flies are mated. The offspring consists of 77 flies with long wings and 24 flies with short wings. Is the short wing condition dominant or recessive? Why? Well, usually we assume that the ones that are more common are going to be the dominant, right? Okay, so that would mean that the short wing condition would be recessive because there are less of them. Okay. What is the genotype of the parents? So if we have, so there's less of them, and I'm sorry, I read that wrong. Okay, is the short wing condition dominant or recessive? So the recessive because if you notice, their parents, neither of the parents had short wings because the parents did not have short wings. So it must have been hidden in their genotype. Okay, so what is the genotype of the parents? So if they were 2 long-winged fruit flies and they ended up with short-winged offspring, then they must carry the short wing gene. So that must mean that they are carriers, which means that they are heterozygous. Okay? All right. Number 3 is a question that you have on your homework or on your page, so I'm not going to do that for you. Okay. Let's see. Number 4, in humans, normal pigmentation is due to a dominant gene, big A, and albinism is due to a recessive allele. A normal man marries an albino woman. Okay, so we know that this normal man has at least a big A. The woman has 2 little a's, right? And their first child is albino, little a. So that must mean that he is heterozygous, right? Because the only way that the child got 2 little a's is because one came from dad, right? So dad must be heterozygous. Mom, she's definitely albino, so she's homozygous recessive. What is the probability of having an albino child? Okay, so you can set up the Punnett square, okay? So you can, if you want, you can set it up. So you have, the Punnett square with dad's big A and little a, and mom's little a, and then, you can do the probability. So what's the probability of having an albino child? Looks like it's 50%, right? 50%. What is the probability of having 2 children in a row that are albino? Okay, so if there is 1 out of 2 chance, so 1 out of 2 chance of the first child being albino, and a 1 out of 2 chance that the second child will be albino, then you simply just multiply the 2. So if there's a 1 out of 2 chance the first child will be albino, 1 out of 2 chance the second child will be albino, then for both of them to be albino, it's 1/2 times 1/2 equals a 1 out of 4 chance. Okay, so you just multiply the probabilities together. Okay. Polydactyly, or extra digits, behaves in a human as a dominant allele to a normal condition, little e. A person with an extra toe, so I already know, big E, marries an unrelated person with normal digits. Okay, so that person's little e. The first child has an extra toe and the second child is normal. Okay, so the first child has an extra toe, but since, the mom or dad, it's little e, must be that. And then the other one's that one, so I know that the parents, the first parent is heterozygous. Okay, so I figured that out. So the first child must be heterozygous because one of the parents is homozygous recessive and then got the dominant allele from the other parent. Okay. The second child is normal, so little e. The normal offspring eventually marries a normal unrelated person. So this child, and produces a child with almost normal unrelated person, and produces a child with an almost complete extra toe on 1 foot. Explain these observations. Okay. All right, so we have polydactyly. So we set up kind of a pedigree here. So we have a person with an extra toe marries an unrelated person with a normal number of digits. Okay, so this person, they got married. Their first child has an extra toe, and the second child is normal. The normal offspring eventually marries a normal unrelated person and produces a child with an extra toe. Explain these observations. Okay, so how do we end up with somebody who, in this case, the dominant allele somehow skipped a generation? Any guesses? Well, this person must have received the recessive allele because it doesn't have the extra toe. So how did this person end up passing on an extra toe gene? Or did he or she? Well, my guess is it was a random mutation. Okay. So this person, it just happened to be, you can't end up with, if the genes of this person can't be passed as polydactyly because he doesn't have the polydactyly gene. Okay, so I assume it was just a random mutation that allowed an extra toe to occur. Okay. All right, moving on to our dihybrid crosses. So some dogs bark when trailing, others are silent. The barking trait big T is due to a dominant gene. A second trait to erect ears, big E, is dominant to drooping. So what are the genotypes of dogs of the following cross? So we have a completely heterozygous erect ear barker. So it's completely heterozygous. So big T, big E, little e is mated with droop-eared silent trailer. So little t, droopy. Okay. So what kind of pups would you expect? So the first thing we have to do is figure out the gametes. So with this first gamete, remember, if we do foil, there's going to be 4 different possible gametes. Okay, so we do first, outer, inner, last. Okay, so if I do first, that's going to be big T, big E, outer, big T, little e, inner, little t, big E, and last, little t, little e. Now this second genotype, there's only going to be one type of gamete produced, because if I do first, it's little t, little e. If I do outer, it's still little t, little e. If I do inner, it's little t, little e. If I do last, it's little t, little e. So there's only one gamete, okay? Possible gamete that's produced, okay? So now I just fill in just this row. We don't even have to do the rest. All right, I'm gonna do capital letters first for all of them, and I'm gonna do T's before E's. So big T, big E, little e. Big T, little e, little e. Little t, big E, little e. And little t, little e, little e. That's it. So now we have to show our genotypes and phenotypic ratios. Okay, so basically all the genotypes are different. Okay, so I just have the genotypes. I can't really distinguish them from each other. They're all different. If I look at the phenotypes, what do I have? This first one, what is it? Big T, big E, little e. So that means it's a barker. And then what does the big E mean? So that's barker with erect ears. Okay. And then this guy, he's a barker, but he has drooping ears with drooping ears. And then little t, he's silent. Let's go on a different line. Silent with erect ears. And then this last one, little t, he's just like one of the parents, silent with drooping ears. I said droopings up there. Oh well, drooping ears. Okay, so there, we gave the possibilities. All right, and then number 2, in horses, the color black is due to a dominant gene, big B, while chestnut is a recessive allele, little b. The trotting gait is due to a dominant gene, big T, while the pacing is a recessive allele, little t. So a complete homozygous black pacer. So we have big B, big T. Oh, pacer is little p, or little t, sorry. So black is big B, and then pacer is little t, is crossed with chestnut trotter. So the chestnut trotter is little b, and then the trotter is homozygous, so big T. Okay. so now I have to figure out the gametes, okay? So with the gametes, remember we do FOIL, so big T, little t is the only possible gamete I can do here. So first, outer, inner, last is always going to be big T, little t. Same with this guy, it's always going to be little b, big T. So the only outcome I can get is big T, little b, or big B, little b, big T, little t. That's the only possible outcome I can get, okay? So what kind of fold can I expect? So it's going to be big B, little b, big T, little t, which equals black trotter. All right. What kinds of foals can be expected by mating two horses from the first generation? So if I mated big B, little b, big T, little t, with big B, little b, big T, little t. So with that, I will have to get, FOIL that out. And with foiling, remember, that's going to result in 4 different gametes. So big T, big T, big T, big T, big B, little t, big T, B, little t, little b, big T, and little b, little t. And I'll do the exact same gametes on this side. Okay, and then we'll combine all of the gametes together. Big B, big B, big T, big T, big B, big B, big T, little t. All the different combinations. Mess that one up. Myself confused. Don't you love dihybrid crosses? I like them when they don't involve heterozygotes, personally. And just be lucky that you don't see any trihybrid crosses. Those involve multiple traits. Three traits or 3 genes instead of 2. All right, so we have them all. And then from there, luckily it doesn't say you have to write out the genotypes and phenotypes. So you just have to fill out the square. All right, so number 3 is on your homework. So I'm not going to do that one for you. All right. Incomplete dominance. So this one, it says incomplete dominance. So remember, with incomplete dominance, we're blending the heterozygotes. So that means that it says right here that in snapdragons, flower color shows intermediate inheritance rather than dominance. So homozygous plants, big R, are red. Heterozygous, big R, little r, are pink. And homozygous, little r, are white. So diagram a cross between a red and white, and summarize the F1 generation. Give the genotypes, phenotypes, and genotypic frequency and phenotypic ratio. Now remember from our lecture, you could do the superscripts if you want, or you could do, instead of the R's and the little r's, you could do like W for white. It's up to you. It gave me the R's and the little r's, so I'm just going to stick with the R's and the big R's and little r's. So I'm going to do red. So big R, and little r stands for white. So we're crossing a white with a red, and we end up with big R, little r. But remember, big R, little r is not red, it is pink. Okay, so give the genotypes. So this, all of them are pink. So we have the genotypes, phenotypes, and then if we want to do the genotypic frequency, remember it's the percent of, or the number of homozygous dominants, so that's 0. The number of heterozygotes, that's 4. And the number of homozygous recessives, that's 0. For the phenotypic ratio, that's the number of dominants. In that case, we have 0. The number of intermediates, 4. And the number of recessives, 0. Okay, so this one's a little different because it's an incomplete dominance, so we do include the intermediates there. In the next question, we have diagram a cross between two F1 plants. So we're talking about the big R, little r. Big R, oops, F. And summarize the F2 results. Okay, I don't know why they give you a dihybrid, so ignore that. I guess they're trying to trick you there. All right, so then from here we need our genotypes. So we have big R, big R. So what was the phenotype there? That would be red. Okay, we have big R, so that's pink. And then we have little r, that's white. What is the genotypic frequency? We have, excuse me, 1 red to 2 pink to 1 white. Phenotypic frequency should be the same. Okay. Number 2, a curly-haired man married a straight-haired woman. Their children all have wavy hair. Is this considered incomplete dominance? So you have curly and straight makes wavy. I'd consider that incomplete dominance because wavy hair is kind of in between curly and straight. So if we have two wavy-haired people marry, what would you predict their offspring's hair type? Okay, so you can choose, any letter of the alphabet. Okay, so if you want to set up a Punnett square, so you could do like curly and straight. Okay, if you want to do the different color, and then the wavy would be CS. So if we had set up a Punnett square of two heterozygotes that have, curly and straight, that means we have curly, 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 straight, and straight, straight. So what would their offspring be? They would be curly, straight, and wavy. They'd have all the possibilities. Okay? So there's 50% chance that they would have the same hair type as their parents. There's a 25% chance that they'd have curly hair, 25% chance that they'd have straight hair. An insect can be brown, tan, or white. Their eyes may be red or cream. The genes are not linked. Okay, so this is a dihybrid cross that has two different genes here. So we have a brown male. Okay, so brown male with cream eyes. So big B, little b with cream eyes, little r, is crossed with a tan female. Oops, I'm sorry. I got that wrong. Delete. Brown male, big B with cream eyes, little, is crossed with a tan female. Big B, little b with heterozygous red eyes. Big R, little r. So we have to set up the Punnett squares. So we know what the gametes are for the first one. So it's basically all of the gametes will be big B, little r. Okay? There's no possibility of any other gamete combination. But for the heterozygote, there's 4 possibilities. There's big B, big R. There's big B, little r. There's little b, big R. And little b, little r. So now we just bring them together. So big B, big R, little r. Big B, little r. Big B, or little b, big R, little r. And big B, little r, little r. Okay. And what type and proportion of offspring will you expect? Okay, so let's look at the offspring. So what's big B? That's brown. Okay, so we have brown. And then big R, little r is red-eyed. Okay, so we have actually 2 of those. All right, 2 of those. Oops. And then here we have, I'm sorry, that's tan, excuse me. Get rid of that. Delete. We have big B, little r. So that's brown, cream-eyed. We have tan, red-eyed. And that's, sorry, this is a B. And then finally we have tan, cream-eyed. So those are our 4 possibilities. Quarter each. All right, so moving on, we have our blood typing system. So one of the best known examples of blood multiple allele is referred to our ABO system. So notice we are going back to our superscripts. So big I represents the A allele and the B allele because they are co-dominant to each other, and then the I, little I, is the recessive allele, or the O. Okay, so I'll do the first one. The second one you have to do on your own. So a heterozygous female with blood type A. So if she's blood type A, that's her genotype. So she's big I, and then I'm going to do a caret for the superscript A, and then she's little I for her second allele. She marries a male with type O. Okay, so his genotype is little I, little I. What possible blood types could their children have? Okay, so here's the genotype of the female. So one of her alleles is and big I with the A attached. The other allele is the recessive. Okay, the male is little I, little I. Okay, so that's how we set it up. Okay, and then you do the Punnett square just like normal. So here comes caret. Yeah, it's awkward with the superscripts. I apologize. I just don't want to go through the whole symbol business. All right, so here are the Punnett squares all filled out. So what possible blood types could the children be? So if we have basically an A allele and an I, what's that blood type? So that's type A. And then we have two little i's, so that's type O. Okay. You can do that one on your own. Okay. And then we have number 3, a boy of blood type A is born to a mother of type O. What are all the possible genotypes of the father? So if mom is blood type O, she is little I, little I. If the boy is blood type A, he has to be A caret and an I. So he got the little I from here, and then he got the big A from dad, right? So what's the other allele of dad? Okay, so there's a couple possibilities. One of it could be another A allele, right? So that's one possibility. What's another possibility? So that's one possibility of dad. He could also be, let me put the other A in. Could he be O? AO? Could he be heterozygous like his son? Sure. Okay. Could he be AB? Sure. Okay. Okay, so there's 3 possibilities. He could be homozygous recessive for A, he could be heterozygous for A, or he could be AB. Okay, so those are the different possibilities. All right, there's nothing else he could be. All right, moving on to sex-linked monohybrid crosses and pedigrees. So with color blindness, we have the following symbols. So notice we have the superscripts we're using. So we have the sex chromosomes X and Y. And then notice that the genes are only on the X chromosome. So we have a normal allele is the uppercase letter, and then the allele for color blindness is the lowercase letter, and it's only found on the X chromosome. Okay, so a normal male, so the normal male is X, and I'm gonna remove the caret, by the way, so I'm just gonna do XB, and then I'm gonna do a Y. So imagine the X is way up there, okay? So he's normal, so he's got an X big B Y, and then I'm crossing him with the carrier female. So the carrier female would be X big B, X little b. Okay, so what's the genotype of the father? He's X big B, I don't put anything on the Y, and then the mother is X big B, X little b. Okay. So this is a normal monohybrid cross, so I don't do any foil. So the male gamete, 50% of his sperm will have an X big B. 50% of his sperm will have the Y. For the female, 50% of her eggs will have an X big B. 50% of her eggs will have an X little b. Okay, and then we do the same thing as boys. So X big B, X big B, X big B, X little b, X big B, Y, and X little b, Y. Okay, so here are our possibilities. So summarize the phenotype and genotype of their possible children. So what's the, and you have to read these carefully. So what is the probability of a colorblind female? Do we have any colorblind females here? None. What is the probability of a colorblind male? This one is colorblind, so 25%. What is the probability of a female carrier? This one's a female carrier, 25%. Construct a pedigree chart of this cross that includes the genotypes of all the individuals. Okay, so when we do a pedigree, remember squares are females, or excuse me, males, and circles are females. Okay, so we have a circle for mom. She's married to dad. Okay. When we do the cross or the pedigree, okay, a normal would be unshaded, so a normal female would look like this. A normal carrier would be half shaded because she carries the trait. A colorblind female would be fully shaded, so she would be all shaded. Normal male would be square, unshaded. Colorblind male would be fully shaded because he has the trait. Okay. All right. A vertical line means they had children. Okay. And then let's say they had, 4 children. Here's all the 4 possibilities. So we have a female, a male, and a male. Okay. Remember the mom is a carrier, so we shade her half. We have a female that's a non-carrier. We have a female that's a carrier, so she's shaded half. We have a male that's not a carrier and a male that has the condition. So he would be fully shaded. Okay, so that's how we would construct a phenotype, or excuse me, a pedigree with all the genotypes. All right. Let's see. Number 2 is on your homework, so I won't do that one. Right? All right. Number 3, if a colorblind man, so let's do a colorblind man. So if he's colorblind, he's X little b y, married a normal, healthy woman whose father was colorblind. So if her father is colorblind, she must be a carrier, okay? Because she got one of her X's from her dad, and the only X he has is a little b, right? Was colorblind. Could they have a son with normal vision? Well, what do you think? Let's look at mom. Where does the son get his X chromosome from? Does he get it from mom or dad? He gets it from mom. Is there a possibility that he can have normal vision? Yeah, there's a 50% chance. Because she carries a normal vision, 50% chance, right? There's a 50% chance she'll give him the normal vision X, 50% chance that she will give him a color blindness, right? Could they have a colorblind daughter? Yeah. Okay, there's, dad's definitely going to give the daughter his color blindness X chromosome. There's a 50% chance that mom's going to give a colorblind X as well. That will combine to give daughter a colorblind daughter, right? So there is a chance. All right, number 4, detective denting of the teeth, defective denting. Why did I say detective? Defective denting of the teeth is caused by a dominant gene D on the X chromosome. The gene causes the teeth to wear down rapidly, and usually only stumps remain by adolescence. If a man has this trait and his wife does not, which of his children will inherit the characteristic? Will it be the males or the females? So if the man has it and it's found on the X chromosome, who will have it in his kids? The males or the females? The females, right? Because it's found on the X chromosome. And who gets the X chromosome from dad? The girls, okay? The boys get the Y chromosome. All right. Let's move on. We have sex-linked dihybrid crosses. So in this case, we have two things going on. We have sex-linked traits and we have dihybrid traits. Okay, so these can get very complicated. So if a man and a woman both with normal vision, so normal vision, that means that they are X big B Y and X big B, okay, wait, and then we don't know X, and pigmentation. So now we have to get into the albinism thing. So remember albinism is big A. Oops, let me write this over here. Okay, so we have let's start over here. We have a colorblind albino son. So if the colorblind albino son is X little b y, and he's albino, little a, little a. So we need to figure out if a man and a woman both with normal vision and pigmentation marry and have a color albino son, what are their genotypes? Okay, so let's first of all figure out the man. Okay, so we know that he is X big B Y. We know that he's got normal pigmentation, but his son is albino, so he must be heterozygous. Let's look at mom. We know that she's X big B X, and then she must carry the recessive allele, making her son colorblind. And then we know that she is normal pigmentation, but she must carry the allele. So we know that what their genotypes are. What's the probability of them having this type of son? Okay, so now we have to figure out the gametes. Okay, so this thing we have to do is, again, we do FOIL just like we normally do. Okay, so we do X big B A, X big B A, so that's first. Outer X big B little a, X big B little a. Inner Y A, Y big A, and then last Y little a, Y little a. And then same for mom, X big B, X big A, X big B, X little big A, excuse me. And then outer X big B little a. And then inner X little b A, X little b big A. And then finally last, X little b little a, X little b little a. Okay? And then we do the same thing where we are going to put them all together. So we have X big B, X big B, X big A, X big B, A, X big B, X big B, X big A, little a. X big B, Y, X A, big A, X big B, A. X big B, Y, X A, little a. X big B, X big B, A, A. X big B, X big B, little a, little a. X big B, Y, big A, little a. Oops. X big B, Y, little a, X big B, X little b, X big A, X big A, X big B, X little b, X big A, little a. We're almost there. X little b, Y, big A, X little b, Y, big A, little a. X big B, X little b, big A, little a. X big B, X little b, little a, little a. X little b, Y, big A, little a. And then finally X big B, or little b, X Y, A. So if we look at this genotype, X little b, Y, A, if we see where it is, this is the only possibility. So there's a 1 out of 16 chance that we will get a boy that is both colorblind and albino. Okay. This second one you are going to figure out on your own. Okay. Okay, and then we go into sex influence crosses. So some genes not located on the X or Y chromosomes are expressed differently in the two sexes, and therefore the traits they control are called sex influence traits, such as pattern baldness is caused by an autosomal allele that is dominant in males because of the presence of high levels of testosterone, and recessive in females because of the low levels of testosterone, which is why more men than women are bald. A heterozygous male is bald, but a heterozygous female is not. A woman will only be bald if she is big B, and not bald if she is big B, or little b. Actually, because of the influence of other sex-related factors, most women with big B never become bald, but rather than their hair becomes thin or sparse. Okay. So a couple in their early 20s have the following alleles for baldness. So the man is big B, and the woman is little b. Remember to solve for both gender as well as baldness. So will the man become bald? Okay, so if he's big B, will he be bald? Okay, so remember, big B, will he be bald? That was a yes. Okay, so he's going to be bald because it says up here it's an autosomal allele that is dominant in males because of the high levels of testosterone. Okay, so will the woman have thin hair when she gets older? So if she's little b, we already know that she's not going to, right? Because she's non-bald. So why do we even need to solve this? Oh, I guess we have to solve that because of, okay, so that's a no for her. Okay, so remember to solve for both gender as well. Okay, so we know that his genotype is X big B, Y, and hers is X little b, X little b. So if we solve for this, we have X big B, Y. Oops. Oops, scratch that. That's wrong. I'm sorry. So what we're going to do is we include the, so he is, I'm sorry, he's X, Y, and then he's big B, and she's X, and she's little b. So let's get rid of that, okay? So that means if we do the gametes, we're going to do FOIL again, okay? So we have his gametes, so X big B, X, and then, you know, X big B will be the same, and then the other possible combination would be Y big B. All right? And then for her, the only one possibility she can be is X little b. So if we put them together, we can have a girl that's heterozygous and a boy that is heterozygous. All right, so what are the chances that their sons will eventually be bald? Okay, so pattern baldness is caused by an autosomal allele that is dominant in males because of their presence. So it looks like he's going to be bald, right? So about 100% chance because he's got the dominant allele. Should their daughters be prepared to wear wigs? It looks like here she's non-bald, right? Because she's heterozygous. No, because she is heterozygous, all right? And it says heterozygous is non-bald. Okay? Number 3, you're going to answer on your own, all right? And then I think this is the last, right? Yes. And then the last part, polygenic crosses. So with polygenic crosses, many traits result from many genes, okay, coding for one trait. So for example, we have our skin color. So you can see here, this is just an example. You know, we have multiple genes. In this case, it's showing like 6 different genes, or, you know, coding for one single trait. So, you know, variations within that, even if you have, you know, you could, or in this case, it's just showing three genes coding for one trait. So within that, you might have all sorts of different possibilities in your offspring. So a woman with a genotype that is, you know, big A, little b, little b, little c, marries a man that is big A, big B, big C, little c, describe the skin phenotypes of the parents, okay? So if we look at somebody who is of this genotype, okay, so basically the dominant would be the dark circle, okay? So she's got one dominant allele, and the rest are recessive. So she would be pretty light skinned, okay? So she would be like pretty fair. And then he would be medium. Okay, so she would be fair. She would be fair. He would be medium. All right. So basically, she's got one dark allele and 5 light alleles. He's got 3 dark alleles and 3 light alleles. So he's right in the middle. She's on the fair side. All right. Their child has skin color between his parents. What are the possible genotypes of his of this child? Okay. So if she is basically, if we're crossing this with this, what are the possibilities of the skin type of the children? So what are the possibilities? So if she's, you know, A, B, C, crossed with A, B, C, what are the possible genotypes of the children? Okay, so in order to figure out what the possible genotypes of the child, we kind of have to separate all of the genes of each of the possibilities. So here we have heterozygous for the A's, okay? So if we look at, if we do a quick Punnett square for the A's, These are the possibilities that we can get. So there's 3 possible outcomes for the A's. For the B's, there's only 2 possible outcomes, right? And then for the C's, there's only 2 possible outcomes. So once we figure out what are the possible outcomes for each of the offspring, then we can write out all the possibilities. Okay. So for the offspring, I know that there's 3 possibilities for the A's. So there's this one, and then we do, all the, the same for the B's and C's. So there's all the possibilities for this scenario. Now we do the different B's. Okay, so here's the A possibilities for the different B's. I already did that one. Oops. Okay, now I have to do the C's. A, C. All right, and I think the only thing I have to do left, did I get them all? Okay, and then the only thing I have to do now is just have and the rest you can do on your own. All right, so that is it for the Mendelian genetics. So basically the questions that we did not answer together, you have to answer on your own for credit. In addition to those questions, you'll be answering the questions on page 164. Let's go all the way back up. Oops, 164. So that's this page 164. And then 162, this page, and then 145. Which is this page, and I did some of it for you, okay? So once you have completed those pages, then you're going to submit those for points. If we take a look at our reviewing for knowledge, oops, wrong one, this one, just realize that we have our dominant traits are capital letters, and we give you the letters that we want you to use, and then the recessive traits are the lowercase letters over here, and then you're going to answer the questions, okay? Pretty straightforward. Over here, make sure that you're listing the gametes, not the genotypes. Show your work. So that includes like a Punnett square and then the genotypes and the phenotypes of the offspring. Okay. Once you're done with the reviewing your knowledge, go ahead and submit that for points. All right. In addition to your lab pages and your reviewing your knowledge, you're going to be submitting your critiques for your student or scientific report workshop. So for the scientific report workshop, you will be given, using basically reviewing 3 students from our scientific report workshop. Sorry, it's hard to see on here because it's in instructor phase. But you'll be choosing, you'll have 3 students that did the same experiment as you, so are working with the same variables. So, you know, if you did salinity, you're going to be critiquing 3 students that did salinity, okay, or, you know, temperature, or vice versa, or something like that, okay? So I'm going to try to stick with students that did the same experiment as you. So you're going to be grading, you know, their reports. When you are grading their report, okay, you're going to be using this checklist. Okay, so this is the checklist that you're going to be using to make sure that they are doing a good job on their report. Okay, so as you go through their paper, you're checking off to make sure that they included these things in their paper. Okay, so if they did not give a title that communicates the subject, then tell them in their critique that, you know, you did not include a title that includes the independent variable. You forgot to include yeast as the organism that was tested on in your title. Okay, so you're going to make sure that, you know, these boxes are checked or not checked. Okay, and then this is what you're going to include in your critique. Okay, so this is, you know, make sure that you're including this, and then this is what you're going to be passing on to your fellow student. Okay, and then make sure, you know, you're writing some comments along the way. So this is what you're going to pass on to your students, and, you know, write some information on there as well. All right, so that's what you're passing on to your fellow student. 3 of these, 3 papers that you're grading for 5 points. All right, if you have any questions, feel free to email me. I know this is a long video. If you have any questions, feel free to reach out.